Number 6, human blood cells. A prepared slice of human blood was observed. Now they are going to give you this. For this, they cannot really tell you to draw your blood, so they are going to give you the image in your question. And they tell you to, number 1, draw and label red blood cells, white blood cells, and platelets. State one function of each cell type. Explain why red blood cells lack nucleus. Now, this is what you are going to be seeing. You're going to see something like this. Now, this red part is your red blood cells. This part, can you see that it has nucleus? So it is your white blood cells. And these tiny, tiny things, this is your platelets. Then this other part, this liquid part, this other part is your plasma, and they make almost all the percentage of the blood. So now we have drawn and we have labeled it. State one function of each cell type. Now we want to name each function of the red blood cells, the white blood cells, and the platelets. Now the red blood cells, their main function is to carry oxygen. Why? Because they contain this hemoglobin, which is the red pigment that is found in these red blood cells. Now these pigments, they react with oxygen to give us hemoglobin. Now these red blood cells are responsible for carrying oxygen throughout the body. We have white blood cells. Now these ones, they act as the fighters. They act as the antibodies. They fight every foreign bodies that are found in the system. Then we have the platelets. This one, they are proteins. They are the ones that are responsible for the clotting of blood. Explain why red blood cells lack nucleus. Now you notice, white blood cells, they have nucleus. But you see, the red blood cells, they don't have nucleus. And this is for a very, very good reason. Now, the reason why red blood cells don't have nucleus is because they need enough space to carry oxygen. That's the reason why. They need to carry as much oxygen as they can. They cannot afford to have a nucleus that will take almost half of the cell, because their cells are very, very small. So that is the reason why they don't have nucleus, because for them to effectively carry oxygen. So you can write that, to effectively carry oxygen. Number 7, test for proteins using the Burette test. Now the question under this part that you can see, a student tested a food sample using the Burette solution. A, describe the steps involved in this test. B, state the color change for a positive result. C, explain what this result indicates. D, state one function of proteins in the body. Let's start with A, describe the steps involved in this test. Now this is the way the test will look like. Your test will look like something like this. They can give you different samples, then they will tell you to put this Burette test. You put them. You separate your samples. Then now in this process, the difference between our Benedict test and this Burette test is that this Benedict test, you have to heat it. But this Burette test, you don't need to heat anything. Just put it there and you'll see your results immediately. You don't need heat energy to initiate the reaction. That's a major difference between your Benedict test and the Burette test. And also that your Benedict test is for reducing sugars, while this test is test for protein. Now after you have put your samples in different test tubes, you can now start putting your Burette, the solution there. Then the one that turns purple, that is the one that has protein. So we normally use this test to test for proteins. Say the color change for a positive result. So a positive result will give a purple color change. C. Explain what this result indicates. It indicates presence of proteins. D. State one function of proteins in the body. So for color, we have purple. For results, we have proteins. And for function, what are the function of proteins? They are body for body building, to repair worn out tissues, and also for enzyme production. So these are some important functions. So out of these functions, they are requesting for one. They can request for one, they can request for two, they can request for three. So these are the three main functions of proteins we have: body building, they repair worn out tissues, and for enzyme production. Number 8, enzyme activity. Now we're talking, this is about enzyme. Don't show you that how does enzyme affect, an experiment. For example, this question, we have an experiment was set up to study the action of amylase on starch. Describe how iodine can be used to monitor this reaction. Describe how iodine can be used to monitor this reaction. Then state the expected result as time increases. Explain the role of amylase. State 2 factors affecting enzyme activities. Now the first question we have, describe how iodine can be used to monitor this reaction. And they said it was used to, the reason they want to see the action between amylase on starch. Now the way we conduct this experiment is that, let's look at this picture. You will notice something. Now in this particular, they will put starch on this, maybe, like, for example, you can take like 3 or 4 petri dishes. You can put in one of them, you can put starch there. The second one, you will put starch and amylase. But you can continue, reducing the amylase or increasing the amylase. So as you are increasing the amylase, it is for different reasons. Now, if you notice something, you see pH 7 buffer. Now, this must happen in a specific pH, a very specific pH and a specific temperature. Why? Because enzymes, they require a specific temperature to perform. They also require a specific pH to perform. Now when you are done with this experiment, you will notice something, that the one that you do not add the amylase, don't forget that we use iodine to test for starch. Now in a normal starch, if you put this iodine, it will be blue-black color, like what we saw when we were testing for the ones for the leaf. Now, but the thing is that as you are adding amylase, as you keep adding more amylase and more amylase, you notice that blue-black color will start changing. It will start changing to brown. Before you know it will change to light brown. Before it's now change to yellow. Now what does this mean? It means that as we are adding more amylase, starch is disappearing. And why is that? Now take an example for your mouth. In our mouth we have amylase. Now why do we have amylase? The amylase that we have in our mouth is called ptyalin. Now this ptyalin, that's what we use to break starch. When we are teaching you digestion, they will tell you that in our saliva we have an enzyme called ptyalin, which converts starch to maltose. So that is exactly what is happening. The reason why the color is changing from blue black as we are adding more amylase is because we are not more seeing starch again. What we are seeing is now maltose. That is how we monitor the reaction. State the expected results as time increases. I've said that you see changes in the color. See, explain the role of amylase. So amylase, now if you want to explain this part, the role of amylase is that amylase acts as an enzyme that converts starch to maltose. So that's what you are going to write in this number. If you are asked that, explain the role of amylase, you just say that it acts as an enzyme that converts starch to maltose. Then state 2 factors affecting enzyme activity. We've said pH and temperature. Now the reason why they're asking me this question, they want to know, just like in your physics, when you do precaution, before you do your practical, you must do your precautions. So they want to make sure that you know the precautions to this experiment. So you can write specific pH, specific temperature. Number 9, structure of a flower. A fresh flower was dissected. Draw and label the petal, the sepal, the stamen, and the carpel. B, state the function of each part. C, which parts are directly involved in reproduction. Now, this is how our flower looks like. So let me label it from this side. If I have to label it, this side is the stigma. This side is we can say this is the style. This is the petal. But we're asked to just label some. Let's just label petal, sepal, stamen, and carpel. Okay, let's just label what we're asked to label. This is sepal. The petals, the stamen, and the carpel. Carpel, the stamen. This is the stamen. Okay. So now we have labeled our flower. The second question is that state the function of each part. Now, after labeling this part, now note that your carpel, when they say carpel, they're telling that asking us for the female part of the flower. The male part of the flower is called the stamen, which consists of the anther and the filaments together. But when you're talking about the carpel, we're talking about the female part of the flower. We're talking about the stigma, the style, the ovary, and the ovules. So when they're telling you to label the carpel, you have to label everything in the carpel. But when you have labeled it, you can now put it. For example, let's say this is my labeling. Then after I'm done with labeling, I'll just do like this. I can now say carpel. Don't just do what I did here as carpel, because they will not be sure that you really know what you are talking about. So first label them, then generalize them as carpel. That's what we are going to do. So now they ask us for each function of what they asked to label. Now the function of the petal, we can say that this petal is used to attract pollinators. Why? For pollination of the flower, so that the flower will not go on extinct. What's the use of the sepal? Now this sepal, when the flower is still in budding, season, it protects the flower itself. Now for stamen, you can say that the stamen, this is the male reproductive part of the flower. Then the carpel, we can say this is the female reproductive part of the flower. These are the functions of those places. then state the function. And for this statement and carpel, you can also go further and take the statement, the part of the statement, you can take them one by one. For example, you can say that for the statement, the anther and the filament, you can say the anther, this is where pollen grains are found. You can say the filaments, this is what holds this pollen grain, the anther intact. Then for the carpel, you can say the stigma, this is where the pollen grains are attracted to. Then for style, you can say this is the part where through the pollen grains travel from the stigma to the ovary, or you can say it is what connects the stigma to the ovary. Then for the ovary, you can say the ovary is what later becomes the fruit. Then the ovary, you can say the ovary is what later becomes the seed. If you do it like that, you have your full mark. And don't forget to always draw with your pencil, and always make sure you label your diagram. Label your diagram. You can say diagram of a flowering plant. Which parts are directly involved in reproduction? The parts that are majorly for reproduction are the male part and the female part. These are our stamen and our carpel. Number 10, breathing rate and exercise. Now this one is just like a practical in a way that you can just even do it where you are. Students measured breathing, and most times it comes as it's a practical, but they don't really expect you to do it. They don't expect you to start breathing out fast, because they expect to know it's a daily life activity. They expect you to know what is already happening. Now, yes, students measured breathing rates before and after exercises. A, record the expected observations. B, explain why breathing rate increases after exercise. C, relate this to energy production in the body. D, name the process involved. Now, from this A, record expected observations. You can say that your expected observation was that before the exercise, don't forget that as a scientist, when you want to record something, you have to have a control. That means that you have to talk about before this, what the activity happened, and after the activity happened. So you can say before the activity or before the exercise, the student's breathing rate was normal. Then later, you can say after, the breathing rate became faster and irregular. You can say faster and irregular. Now that is your observation. Now B, explain why breathing rate increases after the exercise. Now they want to ask that why is it that the breathing rate increases after the exercise? Now if you look at this image, you can see that this is this woman. Before she was breathing, she had a normal breathing rate and a normal heart rate. But immediately she did an exercise, before she was breathing 16 breaths per minute. But after the exercise, she started breathing 34 breaths per minute, and her heart rate became 155. So what's changed? That one leads us to our second question, that we should explain why breathing rate increased. Now, the reason why breathing rate increased is because you know that during respiration tells you that you need glucose plus oxygen to produce CO2 plus H2O plus ATP. Now ATP is energy. Now this ATP is the energy that we need to perform these exercises. It's a cellular kind of energy that we need to perform vigorous exercises. So sometimes now you cannot compare the oxygen that you need to just walk from point A to point B and the energy or the oxygen that we use to run. Now the reason why the person is breathing faster is because the person, you require more oxygen. So because this person requires more oxygen, this person has to breathe in more oxygen. That is the reason why. Now, now if you want to put this in writing, the way you can put this in writing is that you can say that the reason why the breathing rate increases after exercise is because during exercise, more oxygen is used up for the production of energy. So because of now this is why we more the breathing rate increases because more oxygen is needed during exercise. That's a very good way you can put your answer in writing. Now, see, relate this to energy production in the blood. Now, energy production in the blood, we know that in the blood, especially when we are talking about aerobic respiration. Now, this particular thing that I'm talking about breathing rate, it's leading us to respiration. And I've said in every respiration in the body, O2 is also needed. Why? Because sometimes when you have already done your exercise, when you have done exercise without oxygen, something happens. And that thing is called oxygen depth. Oxygen depth happens. And why? That's because the body will not see oxygen to use. Now, because the body will not see oxygen to use, it now used anaerobic respiration occurred. It now gave us lactic acid in the cell. Now, this lactic acid, you want to get rid of it. Now, that is when someone now starts breathing uncontrollably. That is to annul the effect of this lactic acid that has been produced in the muscle cells. Name the process involved. Now, the process involved is respiration, because we have seen aerobic respiration, we have seen the anaerobic respiration. Now, the process of all this is just respiration, and you can further write the equation for the person that is marking to know that you know what you are saying. So this brings us to the end of our class. If you have any question, make sure you drop your question in the comments section, and look forward to our next practical class, because we're talking about the likely questions that will be coming out this year. Make sure you subscribe to our channel. See you in our next class. Goodbye,
Hi, I'm sure you enjoyed the video you just watched. To see more content as that, prepare you for your upcoming exam, make sure to smash the subscribe button and turn on notification. Also, remember that you can use the comment section to send us your questions or your inquiries, and we are happy to take them. I'll see you in the next video. Bye.